So thank you so much for all of you being there, and thank you for that wonderful presentation prior to mine. That was amazing data and so much energy, so it was really fantastic. So I'm going to talk to you today about what a difference an X makes. And as I was thinking about this, and I was just involved even in a symposium today as well as one last week, where I actually think that instead of just the X, it's actually an X. And so some of you might be laughing about this or thinking about, yeah, my X really makes a difference in my life, but that's not really what I'm talking about here. I'm talking really about the sort of the combination between estrogens and the X chromosome. So we're going to talk a little bit more about this as I go on. And those of you who are students, my presentation is sort of a journey from it's how you actually allow your data to drive your career. And that's sort of an unusual way of speaking about it, but that's really what's happened to me. So if there's one thing that I want you to take away from this presentation is that indeed males and females are different. And I think that you actually saw that in the previous presentation, which was really very enlightening. But oftentimes I like to use this slide to basically display how males and females are differently. Forever, basic science research is always focused on utilizing males as the model because males basically have an on-off switch. They're incredibly easy to explore. There's no changes in hormones that make a big difference. And so it's really, really easy. And that's why females have been ignored. Females, I like to think that we're nuanced. We have bells, whistles, knobs, and everything else. And whenever I see data where they're comparing males and females, and then I look at the age range of the females that were included in the study, and those of you who are students, I really want you to start paying attention to this. If you see women in the study that are 18 to 89, you have women of all different levels of hormonal levels. And so you really can't compare them. Women are radically different across all different stages of our menstrual cycle, across as we're aging, across the life cycle. We're different. And that was why the complexity of why women are different is why we haven't been included in any of the biological sciences. And this is really where my career began. And actually my career began as an oops. Some of you might have heard this story, but I actually was, as she indicated, a dietitian. I decided to get my doctorate degree. My very first experiment, I went down, I grabbed a whole bunch of rats. I did a surgery in them. My mentor had a very strong hypothesis about the way the data should end up. We got the data were exactly opposite from what my mentor thought was going to happen. And he tried to figure out what I did wrong. And so we went on and we looked and looked and looked and looked and we couldn't figure out. The surgeries were perfect. Everything was right, except that I had taken the female animals and he expected me to take the male animals. And it was that goof that then catapulted my career to begin to really explore these sex differences. And the reason is that the dogma has always been that outside the gonads, XY and XX cells are functionally equivalent. And even we know from that this is not true. We even know this from geometry, right? We learned this very basic or even algebra, which X is not equal to Y. But in females, the majority of the cells on one of the X chromosomes are silenced. However, there is a portion of those cells on that X chromosome that actually are led to a higher level of production in females relative to males. The Y chromosome, on the other hand, carries most of the genes that are involved in basic cellular functions. I can't think of a better disease to highlight this than COVID. I know that that might sound strange to you, but a lot of the immunological and immune function genes are actually on this portion of the silenced X, so that women actually have a higher level. When you look at the data for COVID, what you'll see is that women may have COVID, but the number of women who die from COVID is significantly reduced relative to men. And so this was just it's been really phenomenal to actually see in one disease that's obviously highlighting and so prevalent in all of our lives right now, there are actually these frank sex differences. They're just amazing. So, and it has to do with what happens between the X and the Y chromosome. But to use a disease other than COVID, because I think we're all probably pretty tired of COVID at this point, let's look at another type of disease that has profound sex differences. Let's look at a disease that actually affects all of us. That is cardiovascular disease. We can all appreciate that cardiovascular disease is the leading killer of US adults, and that's represented on this slide. But what's quite striking is that there are profound sex differences with respect to cardiovascular disease. Men die at a significantly higher rate than women. However, this is becoming a little bit more controversial because when you actually factor gender, and we'll talk a little bit more about gender, that sort of muddies the water a bit. But suffice it to say, the data have routinely indicated that men die of cardiovascular disease sooner and more frequently than do women. So I want you to hold that thought. So I started my basic science career at University of Cincinnati, and then was recruited to UT Southwestern Medical Center in Dallas to really begin to interrogate what it was about these differences in diseases that I felt what was driving these differences in diseases. And I focused my attention on estrogens. And why did I do that? Well, that's because estrogens had been proven to have a function on a whole host of different types of tissues: the brain, the adipose tissue, the liver, the heart, you name it, estrogens have a role in metabolic function. So I felt that I was picking a very good hormone to begin to explore my scientific career. Once I picked a hormone, I had to pick a receptor. And much to my surprise, I didn't realize that estrogens bound to a whole variety of different receptors. And it's become a really interesting field now. Not only is the estrogen receptor a nuclear hormone receptor, but there's also membrane receptors. It's become quite muddied as well. But I had to pick one of the receptors. And this is one of the things that in order to get NIH funding, you have to do. You have to be really laser focused. So I decided, okay, I need to develop a receptor that I want to focus on, and I have to have a rationale. So I dove into the literature and found this New England Journal of Medicine paper, which demonstrated, this person actually lived in Cincinnati, that this human, this man who lacked the estrogen receptor alpha, actually had profound cardiovascular disease. So I decided this is my receptor then. This is a estrogen binding receptor, and it not only appears to have an impact on women, but clearly it has a clear impact on men. So I felt this was a good sign for me to be able to begin to interrogate. So at UT Southwestern, I decided I would start focusing on the brain, and I looked at where the estrogen receptor was located in the brain. Those of you who are medical students or not basic scientists, it doesn't much matter. I use this fancy Krelok's method in order to be able to knock out the estrogen receptor in different portions of the brain. We published this in Cell Metabolism and demonstrated that this estrogen receptor was profoundly important in the brain, in different brain regions, to drive food intake, energy expenditure, body weight, exercise, you name it. This estrogen receptor is critical. And again, we did it in a very, very selective way. So we were able to then compare the differences between the males and females. And it was really, really fascinating. But I wasn't necessarily satisfied with that. I mean, this was interesting information, and it was all done in a mouse. But remember that my background was a nutritionist. So what I really wanted to know was, what if you took a male and a female and you put them on exactly the same diet? We all know that cardiovascular disease differs based on sex. Does it differ based on sex because there's something different with respect to metabolism or nutrition? And again, that's my background. So what I wanted to do is take my males and females, give them a McDonald's-like diet, and see whether they were the same cardiovascular metabolic outcomes when they were exposed to exactly the same diet. So we took this diet. This diet basically tastes like cookie dough. My postdocs, graduate students, and we all love it. It's really quite tasty. The mice love it as well. And so what we were first able to do is we were able to give our cookie dough diet, which is basically, the formulation is like you're having a Big Mac and a Coke, basically. So it has very much human relevance. And so we gave it to our male animals, and we were able to demonstrate that, lo and behold, just like everybody in the world had published, you give men a high-fat diet that tastes like cookie dough, and they gain weight. Even my graduate students did as well. And so then we were able to, with animals, we're able to interrogate the tissues, and we took out the tissues. This is demonstrating the hypothalamus. We took out the liver, the adipose tissue. We interrogated the entire animal. And in all tissues, we found that there was a profound increase in overall markers of inflammation. And it had been well established that inflammation is associated with cardiovascular disease. So this was very well established, and we basically replicated everybody else's data. So that's nice. You give males a high-fat diet, they gain weight, they get inflamed, they get cardiovascular disease. Okay. But what about the females? So we took the females and we put them again on the exact same diet. We had the males and females gain the same relative amount of, or the same amount of body weight. Now they start off at different levels, which is what you can see from my slide. But what I'm trying to show you here is that the animals gained exactly the same amount of weight on the diet. So these data are independent of weight gain. You could say, well, the females gained less. Nope, they didn't. They gained exactly the same amount of weight. So here we have females who are gaining weight on a high-fat diet. What happens to their tissues? And what we found was that the female tissues did not become inflamed. No matter what tissue we interrogated, the female tissues did not become inflamed. Now, at this point, we published this paper and we got a lot of press on this paper. And a lot of the people who were interviewing me said, okay, well, why is that? And if you think about it, teleologically, women have to gain weight in an incredibly healthy way. We do that so we can have babies, right? We have to gain, and if we gained weight and we became highly inflamed, it became unhealthy, that wouldn't be very, very satisfactory with respect to overall reproduction. So those of us who are women on this conference understand that we gain weight beautifully. We do it really easily. We all struggle with it. We gain it, it happens. But what I want you to understand is that we actually gain weight and we're really healthy, even though we are gaining weight. So I had some people say, does this mean that women can go to McDonald's and have a Big Mac and a fry and be healthy? Probably. Is that a very good health message? Not necessarily, but at least what I'm trying to say is that there is something different that happens in our body when we compare it to males. So I wanted to investigate that a little bit further. So one of the things that was elevated in this diet that I gave my mice was actually palmitic acid. It's the most common fatty acid found in animals. It actually also causes activation of the inflammasome, or activation of inflammation. So one of my reviewers said, okay, that's great. So you give the animals the same diet, but what happens in the tissue? So you assume then that males and females incorporate these fatty acids into their tissues at the same rate. That would be what you would expect. But lo and behold, that is exactly not what I found. I found that when males and females gained exactly the same amount of weight on exactly the same diet, the incorporation of the fatty acids was completely different within their tissues. Now, this is just looking at palmitic acid. So the male animals had significantly more palmitic acid, and I believe the palmitic acid, and I was able to show it in cell culture and the like, and I won't bore you with all those details, but it was the palmitic acid that was so much higher in the males that was driving that inflammation and the cardiovascular impact. We even looked at the heart, we looked at everything, and so suffice to say, the palmitic acid is doing this, and the females didn't have the same level of rise in palmitic acid. We went on and did a deep dive. We did an entire lipidomics study. And again, what we found was that males and females, when they eat the same diet, gain the same amount of weight, how they incorporate those fatty acids into their tissues, completely different. And so as a nutritionist, I sat back and I thought, oh my gosh, yeah, I always give people a diet and I never have broken it based on sex. Sure, I'll change the calories, but I've never looked at the macronutrients or the fatty acids and differed those diets based on sex. And so I became extremely interested about maybe overall metabolism, how we actually process nutrients differs based on sex. So just hold on that just for a minute and I'll come back to it in a second. While I was doing these experiments, and the previous speaker even alluded to this a little bit earlier, the National Institutes of Health was looking at all of these data that were coming in and saying, hey, we got to stop all of this. All of these preclinical data are coming in on males. We recognize that sex is really important. So now, in order to get a study funded at the National Institutes of Health, as of 2016, you have to include sex as a biological variable. So let's go back. We started with cardiovascular disease, and we were able to see that there were these profound differences in cardiovascular disease. And so one hypothesis, and my hypothesis walking into these experiments, was that estrogens was a critical driver. So what's been shown now with respect to estrogens? Well, estrogens, the incidence of cardiovascular disease and morbidity and mortality is significantly lower in premenopausal women. And that's what those graphs showed you a moment ago. It's often been thought that the female protection from cardiovascular disease disappears after menopause, implicating both age as well as estrogens as part of the cardioprotection. However, there's more data coming out that even low estrogen in young women is actually associated with cardiovascular disease risk. And there's more and more and more data on this. And again, I was in the symposium, and as soon as I'm finished with this, I'm going to run to another symposium in the airport, but that's another story. Anyway, that shows that it's this really low estrogen, even in premenopausal women, that drives cardiovascular disease. Even data such as early menopause is associated with an acceleration of cardiovascular disease. And this study, and I think that this is really going to change the way we actually begin to interrogate women even at the OBGYN clinic. This shows that women who have irregular menstrual cycles have an increased risk of cardiovascular disease. So I found that to be really interesting. And again, in this symposium that I've been involved in, they're starting to make sure that all women are asked the question, are your menses regular? Because those individuals who have a very irregular menstrual cycle, we should begin to start screening those individuals for cardiovascular risk. So again, what we see is that cardiovascular risk also increases as we age. So it increases because there's lower estrogen. Also, there's probably an age component. I show this slide because I have absolutely no idea how this person ever published this picture. I've evidently, as I'm aging, my estrogen is sinking into my feet. It just cracks me up. So anyway, this is just to show you that our estrogen levels change as we age. There's so much, we could have hours of a discussion about what to do with hormone replacement in postmenopausal women, but to suffice it to say that what the summary of the data would indicate that if you catch a woman prior to going into menopause, in that perimenopausal time frame, you give her estrogen, she's protected from cardiovascular disease. If she goes through full-blown menopause and then you give her estrogen, you actually enhance it. So there's a timing window of opportunity where estrogen makes a difference in a postmenopausal woman. Where do estrogens have their effect? Well, not only did I show you that they have an effect at the level of the brain, but it's also they have a level of effect specifically at that same estrogen receptor in endothelial cells. They modulate inflammation. Estrogens through that estrogen receptor have this critical impact on overall cardiovascular function. So let's go back. So as I was seeing those amazing data with respect to nutrition and its sex differences, I was then recruited to Cedars-Sinai Medical Center. And Cedars-Sinai Medical Center is in the middle of West Hollywood. It's right in the middle of where you see all the stars, which will be really important to my story in a moment. And so I was actually recruited there because they wanted me to do translational medicine. Those of you who are students or basic scientists, you might recognize that translational medicine is the cat's meow. It's what we do is we take basic science and we move it into the clinic. It's really hard. So that's just a little aside, but it's actually really hard to do. So anyway, I was recruited there and off I went. And I tried to figure out how am I going to, because I can't interrogate humans the same way that I do mice, how am I going to replicate what I did in my mice to see if there's actually a human component. And I decided to utilize this approach, and that is that I can take stem cells from humans, I can take them from men and women, I can take them from obese individuals, lean individuals, and I can put them in a dish and I can grow those cells and I can transform them into, in my case, I transformed them into a neuron. And this was sort of the state of the art. And I was like, this is going to be so incredible. I will take human cells, turn them into a neuron, and then expose those human cell neurons exactly the way that I did in my mice and be able to replicate all of my findings, and I'll be off and running. So I took those cells of humans, put them into the dish, and got exactly opposite the results that I expected. And I was like, wait a minute. Now what? So then I said, okay, these preclinical results are completely different than my animal results. Is the problem humans aren't mice? Well, that's true, but that's pretty frustrating because there's an awful lot of my data that was generated in mice, and that would mean that everything is bogus. So I really wasn't happy with that. And then I asked the question, is there something that happens to my cells when I take them out of the human, I put them into this dish that might change the overall function of the cell? And then I asked the question, what about this cell culture media? Now, some of you maybe have no idea what cell culture media is or what cells are. So this is just a quick picture. We utilize cell culture for drug screening. This is the first pass. So we take cells either from a dish or from a established cell line, but in this case, I'm taking them from a human, I'm putting them into a dish. In that dish is a whole bunch of nutrients, there's growth factors, but there are hormones galore. And I was like, oh, there's hormones. What are those hormones doing to my cells that are in the culture? So at this point, I'm walking home, I'm scratching my head, I'm pretty darn frustrated. I'm like, what am I going to do? And I run across, okay, I'm sorry, I have to stop here really quick, and then it'll make sense. So the correct term for male and female is actually cis. So a cis female is actually a female who has two X sex chromosomes, and those X chromosomes are living in a hormonal milieu where estrogen is greater than testosterone. A cis male is an individual who's living in a hormonal milieu where the XY cells are living in a milieu where testosterone is greater than estrogen. Okay, so now let's go back. So I'm walking back from my lab, I'm scratching my head. I'm walking past to my apartment, and I walk past this restaurant, which actually happens to be the Kardashians' favorite restaurant. And I will tell you that the paparazzi precede the Kardashians, so don't think any of those pictures of the Kardashians are like, ooh, surprise. They know exactly that there's going to be a paparazzi there. And lo and behold, I run across this individual. This is actually Caitlyn Jenner, formerly known as Bruce Jenner, the amazing Olympic athlete. So I see Caitlyn Jenner and I stop and I say, oh my gosh, what I had just done was I had taken my male cells, my XY cells, and I put them into this dish, this culture media dish loaded with estrogen. I actually had performed a Caitlyn Jenner-like experiment. So let me explain that to you. So Caitlyn Jenner is actually a transgender woman. And so this transgender woman has an XY sex chromosome, but that XY sex chromosome is now living in a hormonal milieu where estrogens are greater than testosterone. That completely changes the whole functionality of those cells. But does it? Do we know enough about what happens with those cells? And so a transgender man, on the other hand, is an individual where testosterone is greater than estrogen. So at this point, I stopped and I thought, ooh, what's happening with respect to gender, transgender? Is this influencing cardiovascular health and metabolism? What's known about this population? I will remind you that sex does not equal gender. There is a small overlap if you're looking at a Venn diagram, but when we talk about sex, we're talking about those sex chromosomes. When we're talking about gender, we're talking about how people feel. It's a socially constructed role. Gender is on a spectrum. You can have a very masculine female, you can have a very feminine male, but what is gender doing to metabolism? What is that feeling of masculinity or femininity doing, and how does that impact on metabolism? Do people who have cardiovascular disease that are women, are they the masculine women? Do we have any data on this? We have none. And so I really want to implore us to begin to start incorporating gender as a construct. Gender identity in the United States, there's an estimated 1.4 million, and it's growing exponentially almost daily with individuals who identify with a gender identity disorder. And this is just looking at age across individuals. There are some phenomenal podcasts of mothers who have transgender children, who the child at age 2 or 3 will come up to the mom and say, Mommy, I'm in the wrong body. These individuals know, even before they've been socialized, that their gonads are not consistent with where their gender identity is. Even if you do fMRI studies with these individuals, their brains light up like their gender identity, but not their gonadal sex. It is phenomenally interesting. And so what these individuals then do is then they try to seek to align their gender identity with their overall body. And the way they do this is by doing some level of hormonal changes. And so there's now a whole host, I'm sorry, let me go back. There's a whole host of data now with different organizations who are providing this prescription to be able to align the gender identity with these individuals. And for a transgender female, what we do for these individuals is we give them estrogen and we suppress their testosterone. Now let's go back to cardiovascular disease. So we know that in a man, we have an increase in cardiovascular disease risk. We know that in women, there's lower cardiovascular disease risk. So would you not predict that if you're a transgender woman, you would actually be protected against cardiovascular disease? Here I've just set up the stage that estrogens are protective. We're taking a man, we're giving him estrogens to align his gender identity. So wouldn't he be protected? She be protected from cardiovascular disease? And the answer is no. And at this point, I was completely baffled. remarkably, the second leading cause of death in transgender women is cardiovascular disease. I was struck. And so if I look at this graph as well, this is even compared to cisgender men, cisgender women, or transgender men. There is something unique about an individual who is a transgender female. When you give her estrogens, she has an increased risk of cardiovascular death. I was really, really confused. So I thought, well, maybe it has to do with age. And this is just to remind us that as a country, we are going to see more and more individuals in the aging community. And so I thought, well, maybe this has to do with the fact that these transgenders are actually just older, and that's why there's an increase in overall cardiovascular risk. And indeed, that's not the case. As a matter of fact, these transgender individuals won't even make it to some of these later stages in life because they're dying so quickly of cardiovascular disease. So transgender women are dying faster than the general population. And what's the biggest driver? Well, they do have HIV, they have suicide, but cardiovascular disease is right up there. So what is it? What's going on with that? And so one might say, well, it's because they're getting estrogens, and estrogens might cause emboli, and there might be some problems with giving estrogens. And indeed, yes, they have higher levels of stroke and myocardial infarction, as well as venous thromboembolism, but that's mostly due to the older studies where they were giving them oral estrogens. Now, when they're giving them transdermal estrogens, you're not seeing the same level of venous thromboembolism. So that's not necessarily why these individuals are dying of cardiovascular disease. So I then thought, let me explore this a little bit further. Let me understand more about this cardiometabolic health in transgender women, because not only is this a growing population of individuals, but it will also enlighten us about what happens in the cis population as well. We're confused. Is it estrogens? Is it aging? Is there something else going on? So let's begin to sort of utilize what the knowledge is that we have to explore this a little bit further. And so I was diving into this gender data, and someone came up to my office and said, I understand that you're interested in transgender individuals. And I'm like, yes. And they're like, well, we have a data set that you can have. I'm like, a data set that I can have. Here I am a basic scientist. I'm given a human data set of transgender individuals. So I thought, how cool is that? Well, my transgender individuals, actually, when I first graphed the data, had this enormous discordance. It was just like there was nothing about them, the set. It was just a mess. And I'm like, why is this a mess? And so I looked at the transgenders that I actually had, and some of them had had gender affirming surgery. So for my population, these were transgender women. 8 of which elected to keep their testes, and 4 of which who had elected for an orchiectomy, which is a portion of the transgender affirming surgery. So they removed their testes, removed the source of testosterone, and therefore they were becoming more feminized by that population. So here I have, it's a very small subset of individuals, but it allows me to begin to interrogate the system a little bit more. So they came in for 2 visits. They came in for an oral glucose tolerance test. They also came in for an MRI. So we looked at their liver and looked specifically for NASH or fatty liver. And when we looked at the data, we were struck. Now, I have never had animal data that look as beautiful as this. So here it was, there are super different, huge differences. So those individuals who had elected to keep their testes were profoundly insulin resistant. Those individuals who elected for the orchiectomy had normal insulin sensitivity. And even if you look at the fasting insulin levels, what you can see is that those individuals who had elected to keep their testes were profoundly insulin resistant, even with their fasting levels. Now, would you have a priori predicted this? In actuality, you would have not, because there is a whole host of data that say in men, and remember, these are transgender women, so they have an XY chromosome, lower serum testosterone is associated with insulin resistance. Here, my individuals who had low serum testosterone were insulin protected. Those individuals with high testosterone were the ones who had insulin resistance. Again, there's something that flips in these transgender women. So this is just looking at the data. These are the individual data points. Those individuals where the arrows are, those are the ones who were profoundly insulin resistant with a high testosterone. When you look at this is just showing you the data as well. So when we looked at the hepatic steatosis and we looked again at the differences between the two groups, those individuals with testes profoundly had amazingly fatty liver. Those individuals who elected for the orchiectomy had no fatty liver. This becomes really important because the insurance companies, except for Colorado, which I think was just on NPR 3 days ago, now is going to mandate that the insurance companies allow transgender individuals to be able to afford this gender affirming surgery. Prior to that, it was massively expensive and only a few individuals were able to have this operation. Many individuals actually went into the military prior to the previous administration because this procedure was covered under the military and now it is no longer. So again, what our data are saying is if we don't allow these individuals to have this surgery, we're putting them on a life course of potentially cardiovascular risk. We also looked at, so this is just looking at the testosterone ratio again, and those individuals with the highest testosterone had the greatest amount of hepatic steatosis. We also looked at body fat distribution, and we said, okay, the hypothesis would be that if you take an individual with an XY sex chromosome and now you give them estrogens, you should see an increase in overall fat tissue and a change in overall fat distribution. And to our surprise, what we found was that those individuals, again with the highest testosterone, exactly opposite of what we would have predicted, had the highest amount of body adiposity and the most amount of visceral fat, but also a large amount of subcutaneous adipose tissue. Now, I know that a lot of you are very smart and well astute, and you're thinking, okay, she's using the words testosterone, but we know that testosterone actually gets converted to estrogen. So is she really looking at testosterone, or is she actually looking at the precursor to estrogens? Well, I can tell you that we actually looked at free testosterone, and it was the testosterone. It was not, and we also looked at SHBG. We looked at all of the different forms of testosterone, but it actually was the testosterone, the free testosterone, that was actually driving some of these cardiometabolic risks that we saw in these individuals. Much more exploration can go into this conversion mechanism. So at this point, I stopped and I said, okay, this suggests to me that there's an optimal ratio between the testosterone level to the estrogen level. And is there an optimal T to E ratio? And does that then inform the sex chromosomes with overall cardiovascular risk? So instead of a lot of us just look at one thing, it's either estrogens or nothing, or it's testosterone or nothing, but we forget about the fact that there's a ratio of them. And even as we go through our lifespan, there's a ratio. So at different peaks of our life, we might have more T and less E. And now the data are even coming out very, very strongly to suggest that as women get more T, that's when we have that increased risk of cardiovascular risk factors. So this is just, again, data that's been published on the T to E ratio. There's even a funky little calculator that you can go online and get. I don't know why you'd need one, but it's just T to E. But anyway, there's a calculator. And this is just relatively new data, again, demonstrating in women, much like in my transgender women, those individuals, those women who have a high T to E ratio are the ones that have the highest cardiometabolic risk. And so this is just, again, looking at in my data set, again, those individuals with the highest T to E ratio had the greatest amount of cardiovascular risk. So what about the actual tissue levels? And so we had done a study a long time ago where we actually took adipose tissue and we were given money by an organization to ask a really silly question. And that was, is male adipose tissue the same thing as female adipose tissue? And so we went in, we looked at the adipose tissue, we interrogated it, we did a whole omics study, and we found that female adipose tissue is nothing like male adipose tissue. And this is one of my colleagues who kind of went in and did a replica of that study where he went in and looked at muscle from males and females and did transcriptomics and profiling. And again, what they found is that male and female muscle are not anything alike. Now, let me step back to the adipose tissue. After we had published these results, I then got invited to go to a plastic surgeon conference. And the plastic surgeons were kind of up in arms because forever they had been transplanting male adipose tissue into female face and never ever thinking about the fact that, oh, the adipose tissue, even looking at the profiling of the adipose tissues, the gene expression profiles, they're not the same. What happens when all of a sudden I put male adipose tissue into a female face? How does the adipose tissue work? How does it function? Does it take up does it become more inflamed? Lots of information that they hadn't even been aware of, because again, it goes back to that basic premise, the dogma. Once you are outside the gonads, tissue is the same, but it's not. But this one's really interesting with respect to the muscle. So this is just, again, looking at a Venn diagram where we're looking at different gene expression profiles up and down between male and female muscle. But why this is so important is that one of my favorite stories is this. And I actually read this even before I was a basic scientist, way back in the day when I was a dietitian and I was a huge runner. And I picked up Runner's World, which is sort of the gospel according to runners. And I found this article, and I was so influenced by this article. So it's sort of the premise is, ever wonder how much faster or slower you'd run if you were the opposite sex? So here's this incredible story, and I call it Janet's story. This is a story of Jim. Jim is a 5'11", 148-pound middle-aged man in excellent condition. And he used to go to this one park or area in Los Angeles. He had a whole group of buddies that he ran with, and they were like gazelles. They were super, super fast. But at some point during Jim's life, he decided that he actually was not in the right body, and he decided to go ahead and do gonadal or gender affirming hormone therapy. So now Jim has turned into Janet. But now as Janet, same body habitus, so still a 5'11", 148-pound middle-aged woman in excellent physical shape, same running body, same everything, same running regime. But now as Janet, she is more than 2 minutes per mile slower than she was as Jim. So just think about that. This individual, same body, same everything, but now just with using the hormone, just changing the hormonal milieu with respect to what's being communicated with those sex chromosomes, has profoundly impacted this ability of this person to run as fast as they were able to run before. And I was just blown away by this. There's something really interesting about this sex hormone to sex chromosome that changes the functionality of these tissues. So not only is this profoundly interesting with respect to this individual or the transgenders in general, but what about transplant? So I was asked to give a presentation at one of was the renal transplant committee national meeting. And they asked me the question, what happens if all of a sudden you take a male kidney and you put it into my body? Again, think about that. So you're taking a male kidney that has an XY sex chromosome. Now you're putting into a hormonal milieu where estrogens are greater than testosterone. What's the functionality with respect to that kidney? Have the transplant surgeons been paying attention to this? They haven't been. The data on this is really, really thin. And I urge those of you who are medical students who are listening to this presentation, start to pay attention to things like this, because this is a phenomenal study. You can fall off a log. All you have to do is just go in there and look at the data on this, and you'd have like a New England Journal of Medicine article, because we need to understand what's happening when we're doing these transplants. What happens if I get a liver or adipose tissue or kidney from someone who is a different sex than I? Then one of the transplant surgeons even asked me another question. They're like, okay, so say you rejected the male kidney, now we transplanted a female kidney, what happens? Now, so it's almost, I don't know. There aren't any data on this, but it's really, really important. And I think even we just had the Olympics, and as we're listening to a lot more of sort of the Olympic organization, how do we deal with individuals who have different sex chromosomes, and what do we do about the ratio of those sex chromosomes to their sex hormones, and how does that impact their overall level of performance? These are big, big questions that haven't necessarily yet been addressed. So what are the summaries of what I've talked to you about so far? So in my preliminary data with the transgenders, that suggests that the transgender women who actually opted for the bilateral orchiectomy had improved metabolic health. And I think this is really important because, again, this is not an option that's afforded to many of these transgender individuals. And I'm very, very concerned if we don't learn more about what we're doing. It's almost as if we're fighting against their endogenous hormones, and that's having this impact on overall cardiovascular health, but more needs to be studied. Additionally, those individuals who retained their testes were stratified according to their testosterone and had the greatest hepatic steatosis and insulin resistance. So we are all striving to have personalized medicine, and that's really the goal with respect to all medical care. We understand that one size doesn't fit all, but when we look at health, is medicine personalized by or is health personalized by sex? The answer is no, and this is just looking at the number of publications that have been out there where we actually are focusing on health, sex as a predominant factor in health. And so again, I implore all of us to begin to really start to focus on this, because I think so much more will be learned, and we'll be able to actually enhance the health care of not only the men, women, but also those individuals who have gender dysphoria or are the transgender individuals. So with that, I'd just like to thank all of my collaborators with this. It's been an interesting ride. Again, my data has driven my career and my passion. And so I hope that those of you who are students, that you'll listen to your data and allow them to take you where you want to go. So thank you very much, and I'm more than happy to take questions. Thank you.